Okay, thank you all for attending my session today. This talk was actually originally supposed to be given by Thomas Neumann, our CEO, but he couldn't make it here, so you're stuck with me. But today I'll be talking about biofabrication of the kidney proximal tubules, and so I'm going to showcase a few of our other organ structures and some applications that we work on at Nortis. So with that, I want to give a quick background. So I got my PhD at the University of Iowa's Department of Biology under the mentorship of Dr. Stephen Green. And my area of focus was actually not in microfluidics, organ on chip, none of that. Actually, it was in hearing loss research, noise-induced hearing loss. And my thesis was in the pharmacological regeneration of the synapses, the connections between the inner ear and the auditory nerve that connects to the brainstem. But some of the skills that I sort of developed during my PhD were these complex in vitro, 3D in vitro structures. So this is a neonatal rat cochlea, and I actually took this image with an iPhone through the eyepiece of a dissecting microscope, which was very difficult, but we managed to do it. And some other skills you sort of develop when you do this kind of work are advanced imaging. And as you will see over the course of my talk, our technology is very amenable to imaging. So that's something that I'd like to showcase. And it all worked out pretty well for my organ-on-chip introduction. So having said that, I want to go over quickly the drug development process and sort of where we fit in that whole picture. And I apologize to the people who were at the educational session this might be a bit of repetition for, and also for you clinical translational scientists out there. This is an oversimplification, but I think we'll get the gist of what we're trying to do here. So at the beginning, we sort of have the basic research and drug discovery preclinical phases, and what are the kinds of things that we're looking for? We're looking for pathobiology of our molecules, what the biological mechanism is, whether we're hitting the molecular targets of interest, and from this we manage to come up with some lead molecules that we select. And then if all that goes well, we can advance to the clinical phases of our study where, again, we're sort of looking for similar things in humans. We're looking for safety and toxicity, proof that you're getting the mechanism that you saw in the preclinical phases and the clinical stages, and also whether you're hitting your molecular targets. And then if that all goes well, then we get our regulatory acceptance and move straight to market. It's nice and easy, right? The problem is that when you get from this stage to this stage, as we all know, most of the drugs tend to fail in the early stages of clinical trial. And that is largely due to something that happens here that we cannot translate. So there is not enough good predictive technology that allows us to see what's going to happen in the humans. So typically what this looks like is in our in vitro setting, we might have a good result in preclinical stages, and then we move on to our furry friends, and our rodents will do well, and then a canine might do well, and there's usually a non-human primate somewhere in there as well, and then in early clinical trials we fail. So what is it about this stage that sort of leads to this failure in the clinical trials? Well, let's start off with our in vitro systems, our sort of traditional in vitro systems. You don't have to be a biologist to know that if you take some cells out of an organ and put them in a dish where they're just sort of sitting there, they're not going to do the exact same thing that they would do in vivo. And there's largely one of the many things that is different about this system from what happens in vivo is we lose our cellular polarity. The directionality of all the drug transporters and everything is sort of different. And then given the significant similarities that we have between humans and other mammals, we can't deny that there's a lot of differences too. I mean, dogs can't eat chocolate. I can, and I'm okay. They would die if they did. So we have to take into account the major physiological differences between animals and humans. So this is sort of where our technology and other organ-on-chip technologies come in. We're sort of giving a solution where you can use human cells to address these predictive problems that we're seeing in the preclinical stages. So I'm going to give you a quick tour of our most popular chip here, the triple channel disposable microfluidic chip. I'm going to just go through what these different components on the chip are. So we have a PDMS microfluidic network where these blue squares over here are bubble traps. And in the case of our triple channel chip, we have 3 independent bioreactors. Let's just take a zoom in on one of these. When our chips are manufactured, there is a thin glass mandrel, a fiber optic glass mandrel that is in place in the middle of this red collagen chamber, which would be a collagen chamber after it comes to our biology group. But this is about 125 microns in diameter, and the ability that we have with this mandrel is we cast the collagen gel around this mandrel and then extract it, leaving a hollow scaffold in which you can seed cells. So once we extract that mandrel after we've allowed the collagen to settle, we have cell injection ports in which we can seed cells. And what that would look like sort of downstream is, in the case of our kidney proximal tubule chip, you have a nice 3D structure here. And we hear a lot of 3D tissue culture and 3D systems and everything. This is a true 3D system in the sense that the tissue is not coming in contact with anything but the biologically relevant matrix that surrounds it. There's not one side that's touching an inorganic surface. It's totally surrounded by matrix, and it's continuously perfused. Okay, so how does that perfusion come about? Well, I've showed you how the tissues are seeded into the chips. Well, this is a perfusion module that we sort of plug the chips into, and if you pop the hood on this thing, you'll see that there are media reservoirs, there are source reservoirs and waste reservoirs that have this candy cane structure in them. Okay, and this is what it looks like from the front before you plug the chip in. So in the case of our triple channel chip, we would see we have 3 independent experiments, so we have a source and collection for each of those. And you can fit a 0.5 mil Eppendorf tube in our waste reservoir. So let's say you have a drug you're perfusing through the chip for 12 hours and you want to collect some effluent. You just open this up, flash freeze this tube, put another one in, close it, put it back into the incubator, and then go to lunch. How is the perfusion actually driven in the chip? Well, we use pneumatic pressure using a gas pump. So pressure is applied at this manifold, and we have a precision microcapillary flow restrictor, and these come in different sizes, different inner diameters, depending on how much shear you want to deliver to the chip. So you can have your chips perfusing at the same flow rates and subject them to different shear depending on this flow restrictor. And so pneumatic pressure is applied, pushes down on the media, and the media has nowhere else to go but into this straw and into the chip. And it's a one-pass perfusion, so the tissue sees the media once and is collected on the back end for effluent. Okay, so that's how the module works, and the module is placed into these docking stations that hold 12 chips, and in the case of the triple channel chip, for example, you have 3 experiments per chip, so multiply that by 12, and you can fit actually up to 4 docking stations in a standard CO2 incubator, but here we have 2. It gets a little bit busy if you put more than 2 in. And you can have some chips perfusing while others aren't. You can close the valve on one single tubule on a chip, so it's very, you don't have to have everything going at once, depending on what kinds of experiments you're running. Okay, so now that we know how the system works, a quick word on the kidney proximal tubule, which I'm sure most of you are aware, is this region over here where the highest trafficking of xenobiotics and drugs from anywhere else in the body is seen in this center. So it's a very important place for the decision of what molecules are going to get back into recirculation to blood or excreted into the urine. And how this is done is by a very tight and specific repertoire of drug transporters on the basolateral surface or the apical surface of these kidney proximal tubule cells. And this kind of goes back to the whole idea of a true 3D tissue. It's important to have the transporters in the right place, and you have to deliver your drugs either basolaterally or luminally, depending on what the mechanism is, if you want to emulate what's happening in vivo, right? And a nice example in terms of staining of this is we took a kidney from a human and sectioned it and stained for megalin, OAT1, and sodium potassium ATPase, which are 3 of these key drug transporters. And you can see the expression profile of these drug transporters in vivo from a human kidney section. And then we have a section halfway through the human kidney chip that we offer and the basolateral surface over here. And as you can see, the staining profile is extremely similar to that of a human kidney in vivo. Okay, so we have our human kidney chip now, I kind of want to just say a quick note on our imaging capabilities. Our chip has a 1.5 cover glass on the bottom of it, which is ideal for imaging, and you can get really nice resolution. Why is this important? Not just to make pretty pictures, but you have to be able to tell the polarity and the location of the proteins that you're looking at when you're looking at these tissues. So if we were to image just a little bit lower, about here, so it looks like you're unzipping this tubule a little bit, you can see on the basolateral surface the expression of this zone 1 marker, and then on the lumen, you can tell based on the location of the nuclei compared to where the stain is, whether this is a true basolateral or apical stain, and in the case of zone 1, as we know, an apical surface marker. So very amenable for imaging. And this is important for seeing what the effects of drugs are. If we can deliver drugs basolaterally, so we know that in the case of this is a OAT1 marker over here, which is a basolateral stain. We can deliver them luminally, and so being able to see where things are is very important. Seeing is believing at the end of the day, right? A quick note on a group from AstraZeneca who published a paper using our chips last year, and you can stop by our booth and get a copy of this paper if you're interested. The story here was basically there was a drug that had done really well in preclinical in vitro settings and cultures, and then did well in rodents and did well in canines and non-human primates as well, and then failed in early phase 1 clinical trial. And so basically, long story short of what AstraZeneca's group was trying to do here was to see if we had used the human kidney chip to run the drug through before, would we decide to even send this drug to clinical trials? And the moral of the story was no, because the chip was able to predict the same nephrotoxicity that was detected in early phase 1 clinical trial in the chip. So it's a good predictive model for what happens in vivo. Okay, so we have a lot of different chip configurations, and our chip has a flexible design. We oftentimes get new collaborators and customers coming to us and saying, well, we want to do this. We want to put this kind of spheroid or this kind of organoid or these types of cells, and we need a little more space in the chip. We can kind of rapidly prototype and change things up, but this is just a few of the form factors that we show. And I'm, in a minute, going to talk a little bit about our dual-channel chip, which has some capabilities that you can't do in the triple-channel chip. But a quick word on what are our guiding principles for building the commercial products that we do. Well, we obviously talk to key opinion leaders. It's very important to know what the needs are from key opinion leaders, but we have to address what the customers at large are sort of requesting from us. And you know, we hear sometimes about wanting to make these organ-on-chip systems and these 3D culture systems more high throughput, but that's kind of besides the point in a way, because the high cost of these systems and the complexity has to sort of have an advantage if we're going to use them, and it doesn't necessarily need to be as high throughput as the earliest stages of preclinical preclinical testing. And there has to be a stark comparison to 2D and animal models as well. There has to be a clear advantage. Ease of use. So, you know, a lot of people have an experience of where they go to their favorite organ-on-chip company, buy like 500 chips and very excited to do their experiments, and then they start doing the collagen filling or the hydrogel filling. It's not as easy, they all look kind of different, and the seating all looks kind of different, because there is a bit of artistry behind getting these things to work, which is why we've moved to a pre-seeded chip model, and now we're sending our chips with pre-established biology, currently just in the US, but the chips come with the fully grown tissues in them already, and we have a very tight quality control in our biomanufacturing department, allowing us to send these things to researchers, and they run their drugs, do their experiments, and it's much easier that way. And then it's important for us to not overpromise and also to learn from our customers. We are constantly evolving the products at Nortis based on the limitations that we find from our clients and our collaborators. So this is a ever-evolving process for us. And a lot of our technology is driven, in fact, most of our technology is driven by our academic partners through grants. So what will happen is we'll talk to some specialist. In the case of University of Washington, for example, we have our kidney specialists that we work with a lot and who teach us so much about the biology of what needs to be done. And we drive the chip design based on that biology. But using this collaborative effort, we're really able to come up with the best models. Okay. Now I want to move on to our dual channel chip, which is a little bit different from what I showed you earlier, because instead of one, we have two of these fiber optic glass mandrels in here, which gives us a couple of options of things to do. One option is to put two different, in this case, endothelial cell tubes into the chip so that you can have some sort of anastomosis type thing, or you could have one, as Kathy Young showed yesterday, one kidney tube and one endothelial tube. So there are two options here, but you could also just leave one of these empty and have one cell tube. So you could do experiments where you have, in this case, for example, an endothelial tube, and from the empty channel above, we perfuse something that will promote angiogenesis, like VEGF in this case, and you'll see sprouts growing towards that gradient. If we take a closer look, you can see we have, again, good resolution for imaging. You have to be able to quantify these things in a good way, and we can actually measure the sprouts very tightly and look at the directionality. And we can also 3D render these things. Like a lot of the times our clients come to us with a drug that has a fluorescent tag on it, and they want to know where that drug ends up. So we can actually get down to subcellular structures and tell where exactly a monoclonal antibody drug or something like that is ending up. Okay, so I want to say a little bit about permeability analysis that we do in-house to assess barrier function. So for passive transport, in this case we're looking at a 0.37 kD, just native fluorescein. What we do is we do some live imaging. We perfuse it through, in this case, a kidney tube, and we measure the rate of diffusion of the fluorescein into the surrounding matrix. And based on the speed and time that it takes for the dye to come out of the tube, we can calculate using some in-house software the permeability of the vessel. So this is for passive transport. For active transport, and just keep in mind the size of this molecule, 0.37 kD, right? Here we have a 0.35 kD 2N-BDG, you may have heard of. It's a glucose analog dye. We glucose starve a kidney tubule, and then we perfuse for the same amount of time this smaller molecule through the kidney tube. And what you'll see here is it will saturate the tube, but you don't get it bleeding out into the surrounding matrix because these proximal tubule cells are doing what they're supposed to do, and they're reabsorbing the glucose, right? So this ability to tell between passive and active transport is very important because you need to know whether you just have a tube with a bunch of holes in it, or is the permeability of those cells truly what you're seeing. So that's for our transport. And again, we do a lot of cool work with a lot of collaborators at the University of Washington. I'm currently doing some experiments with Vincenzo Cerulli and Laura Crisa using pancreatic islets, where we took the dual channel chip, a modified version of it, and had two endothelial tubes and have organotypic cultures of pancreatic islets in the middle over here. And in red, you see a calcium reporter. And we perfuse the same glucose analog dye through the endothelial vessels that anastomose to these pancreatic islets. And as you'll see, over the course of 15 minutes, these islets will take up the glucose dye and coming from the vascular anastomosis, and yes, they basically light up with glucose. So, and this is work that's undergoing right now. Some work from Sloan Kettering, one of our collaborators from a very long time, working on cancer spheroids. So this is a tumor microenvironment made with our dual channel chip, just to show sort of a different application where we can fit these spheroid-like structures in the chip. This is work done by Dr. Magda Skubal at Sloan Kettering. So I want to talk quickly about a little bit of work that we're currently doing in our lab, where we're doing a 2D, 3D comparisons across different species in the chip and looking at both primary and immortalized cell lines to see what their performance differences are. And this is work that we're preparing a publication for, hopefully before the end of the year. So we basically grow these cells in regular culture flasks, and then we seed them into our chips or into normal static 2D cultures, subject them to a known nephrotoxicant, in this case polymyxin B, and then we have imaging analysis on the back end and effluent toxicological analysis. And here's the image of just showing the viability of our chips from primary human, immortalized human, primary canine, immortalized, and primary rat, immortalized. As you can see, they can handle different amounts of drug, which is what you would expect. You wouldn't expect them to all behave the same way, right? And when we measure the amount of ATP in the effluent, we see sort of the same thing. In the case of the chip on top over here, you see a nice rise and fall, like you do physiologically in vivo of toxicology for the human, dog, and rat, with starkly different tolerances. And in 2D, as we know, we kind of see this unphysiologically high rise in toxicology, and then it kind of just lingers there with no washout period. So this goes to show the differences in our chips. You can actually get species-specific toxicological profiles, and they are more representative of what happens in each species in vivo. Quick note on some T-cell perfusion that we recently started introducing in our CRO department. This is just a video of some T cells perfusing through, and you might notice that they change direction every few seconds over here, so they go back and forth, and this is just to show that we have tight control over the directionality of these T cells in our system. This is using an in-house peristaltic pump system that we just developed, and here we have the T cells that were stained to show before what they look like and after perfusion what they look like, because there's always a concern with these peristaltic systems that they're going to crush the cells, but we know that they don't do that in the case of our in-house system. Okay, so now that, just to sort of start wrapping things up here, I want to talk about some really cool stuff that our collaborators at University of Washington have been up to, and this is just really fun to talk about. And you might have seen Kathy's talk yesterday. It wasn't about this project, but we have had our chips go up to the International Space Station now, I believe twice, and we're gearing up for a third time. And this is just one of those fun projects that we get to brag about. This is the cool collaborators that we have. And in this case, I'm not going to go into the details of the experiments because I will ruin it. But basically, you know, astronauts have this rapid bone demineralization in space in microgravity environments, which leads to kidney stones because of the circulating minerals. So basically, this was a kidney stone experiment, and if you're interested in that, you should definitely go and find Kathy Jung and talk to her about it. But here is a sort of video of the casing of our chips on the International Space Station, and I believe we're the only organ-on-chip companies whose chips were able to come back down and get data out of them successfully, so that was kind of cool. This is Christina Koch using our chips in the casing over here, and some of the partners that were involved in this. Bioserve are the people who make the casing, the hardware to allow the chips to be perfused in space. Now talk about a niche business. They make hardware for biological experiments in space. There's not a lot of competition in that realm, but it's pretty cool stuff. And this is me very happily standing at the Kennedy Center. Just pretty cool to be able to see a SpaceX launch in person. I want to go again, so I'm going to have to grease however many palms I have to get there, but hopefully it will happen. A quick note on, we have quite a lot of very cool industry and academic partners, and we're always looking for more bright minds to help us out, build our technology and take us to the next level and stuff. So really, we owe a lot of our success in our different models to our great collaborations and customers. And as you can see, we have a very small team. You know, currently our company is around 23, 24 people. The biology team is about 6 people. So, you know, we have a long list of things that we want to do, but only a limited amount of hands to do, but we're rapidly expanding. So do keep Nordis in mind for that. But that's all I have for today, and I'm happy to take any questions.